Hi guys, we're learning about life science and cell biology in a nutshell. Here we have a diagram showing the organelle peroxisomes scattered in the cell. They're little tiny organelles containing catalytic enzymes, um, which help with breakdown of various large molecules containing alkyl chains, as well as toxic molecules such as alcohols and large fatty acid chain containing molecules or lipids, um, helping with conversion of lipids into sugars or carbohydrates. So we have a cell here shown with the nucleus is the large round part predominant in the cell. The actin filament network is very visible here in the fibers. Peroxisomes are the small dots here in the micrographs. Nice pictures. So in the stained cells, you can see the peroxisomes. Here, a fluorescence digital image of an African water mongoose skin showing fluorescent probes target the nucleus in red, the actin skeletal or cytoskeletal network in blue, and the peroxisomes in green dots. In order to carry out their activities, peroxisomes use significant amounts of oxygen in breakdown reactions, oxidative reactions. For example, breaking down the fatty acids, um, or large molecules containing fatty acid chains, as well as alcohol containing molecules. The characteristic of the organelles of using oxygen would have been important millions of years ago, before cells contained mitochondria, where the Earth's atmosphere first began to amass large amounts of oxygen due to the actions of photosynthetic bacteria. Because photosynthesis produces oxygen, peroxisomes would have been primarily responsible at that time for detoxifying cells by decreasing the cell's levels of oxygen, which was then poisonous to most forms of life because oxygen breaks down molecules. Oxidation or the organelles would have provided benefits of the cell of carrying out a number of advantageous reactions. When mitochondria later eventually evolved, peroxisomes may have become less important in some ways to the cell, since mitochondria also utilize oxygen to carry out many of the same reactions, but with the additional benefit of generating energy in the form of ATP or adenosine triphosphate at the same time. Peroxisomes are therefore specialized in carrying out oxidative reactions using molecular oxygen. They generate hydrogen peroxide or H2O2, which they use for oxidative purposes, destroying excess by means of catalases they contain. Peroxisomes have an important role in synthesis of specialized phospholipids required for nerve cell myelination. Myelin is a lipid-rich substance surrounding the axon of nerve cells, forming an electrically insulating layer around nerve cells. The phospholipids are essential for the proper functioning of the nervous system. The myelin and phospholipids, the production of this myelin sheath, is called myelination or myelinogenesis. So, a specific sequence of 3 amino acids near the C terminus of many of these proteins um, functions as a peroxisomal import signal. The mechanism of protein import is distinct from that of mitochondria and of chloroplasts. The oligomeric proteins can be transported into peroxisomes without unfolding. So this is in peroxisomes catalyze the first reactions in the formation of the plasmalogens, the most abundant class of phospholipids in producing the myelin sheath. Or if the peroxisomes have a disorder, these class of phospholipids are not produced, and the neurological disease can occur. Here is shown what the plasmalogists contain as the phospholipids in the myelin sheaths of nerve cells. That's the phosphate group and the fatty acid chains and the ethanolamine head group. So glycerol phosphate backbone used for phospholipids. And plasmalogens contain an unusual fatty alcohol that is attached through an ether linkage. Okay, so we're gonna learn a bit about molecules called ethers, which are a class of organic compounds containing an ether group, an oxygen atom connected to two alkyl or aryl groups. Ethers are so ethers contain an ether group or an oxygen atom. Ethers feature a C carbon-oxygen-carbon linkage defined by a bond angle of about 110 degrees and CO distances of about 140 pms. The effect of the ether linkage compared to the ester linkage has been observed on the structure and properties of lipid bilayers. The structure and water permeability of bilayers composed of ether-linked lipids, which are called dihexadecyl phosphatidylcholines, DHPC. Esters and ethers are organic molecules which both contain oxygen atoms. Both contain the ether linkage, which is the oxygen. Esters, however, have two oxygens, COO. Ether lipids occur in biological systems, primarily plasmalogens, where the first carbon chain is attached to a glycerol backbone via an ether linkage, instead of more common ester linkages where the linkage is to the second carbon. So plasmalogens have been found in all mammalian tissues. Nerve myelins contain 52% plasmalogens of total phospholipids. For example, in ox heart, 45% of the total phospholipids have ether linkages, primarily occurring in the mitochondria. About 15% of phospholipids from human red blood cells are ether-linked lipids, and in white blood cells this percentage is higher. The effect of the ether linkage compared to the ester linkage on the structure and properties of lipid bilayers is of interest because it is a well-localized chemical perturbation that affects physical properties. In order to focus on the specific linkage, studies have been made comparing DPPC, dipalmitoyl phosphatidylcholine, which has two 16-carbon saturated chains attached to the glycerol backbone via ester linkages, with the model lipid DHPC, dihexadecyl phosphatidylcholine, which has ether linkages on both chains. One dramatic change. So we'll continue in the next part. Thank you.